Welcome back to Exploring the Metaphysical with UUON Foundation. Today, we turn our focus to mitochondria, the powerhouse of the cell. Our mission is threefold. 1. To annotate how these molecular structures fold, coil, and expand in real time using our visualization application. 2. To document these dynamic patterns thoroughly, capturing both structure and potential historical insights for scientific reference. 3. To provide validation-ready models, preparing them for sharing with leading research facilities, allowing in-depth exploration of mitochondrial architecture. In this session, we'll explore the inner Cristae folds, their coiling patterns, and how they maximize energy production. Our models are presented in 3 layers: a full wireframe mesh, a fully textured PBR model with emissive highlights, and a secondary wireframe to study internal structural complexity. As we manipulate the visualization, notice how even the smallest adjustments, down to a single decimal point like 0.0110, can create unexpected dynamics. These tiny parameter shifts can open the mitochondrion into a new, stable configuration, what we like to call a comfortable structure, where proteins and internal elements naturally align. Watch as the Cristae fold and unfold, coiling in ways that are consistent with known mitochondrial behavior, yet reveal subtle patterns often overlooked in static models. The matrix inside displays slight granularity, representing the enzymes and DNA that drive energy production. Observe how the emissive highlights pulse along the inner folds, signaling areas of high activity. Each layer serves a purpose. The full wireframe mesh allows detailed structural examination. The textured PBR model communicates energy dynamics visually. The secondary wireframe isolates the folds and internal complexity, highlighting subtle patterns invisible in a single static representation. By experimenting with folding, unfolding, and parameter adjustments, we can see how the mitochondrial structure responds in real time. Each minor tweak can produce new arrangements that provide insight into the energetics and stability of the organelle. This approach not only illustrates known biology, but also opens doors to exploring hypothetical or rare structural conformations. These models are fully compatible with standard 3D visualization software, allowing researchers, educators, and enthusiasts to integrate them into simulations, presentations, or educational tools. Whether studying energy production, protein alignment, or structural dynamics, this visualization offers a unique lens into mitochondrial function. As we continue, note the interplay between structure and energy activity. The coiling Cristae, the pulsing matrix, and the folding-unfolding dynamics all demonstrate how even minute changes influence mitochondrial behavior. Our goal is to provide a real-time, interactive exploration that captures both the complexity and the elegance of these molecular powerhouses. Stay tuned for more organelle visualizations in future sessions, expanding your 3D library and deepening your understanding of cellular architecture. By documenting these dynamics now, we create a bridge between computational modeling and biological insight, ensuring that when these models reach research labs, they are ready for immediate exploration, analysis, and inspiration.